Okay, so today we technically our last lecture, um, actual lecture with new material. So we'll cover the antibiotic resistance. We may have a few slides remaining, um, not too many, and then I'll finish that next week. And then next week also, as per our earlier discussions, we're gonna have a sort of like a review week, or rather questions and answers, Q&A session. So the way we're going to structure this is I'm not going to go over all of the lectures, or I guess not all of them, but the last couple of lectures that we covered in the course, because everything is posted. There's no need to be doing that, but instead it's going to be, in addition to maybe covering a couple of slides that I may not finish today, we will have students show up with questions regarding the material, the last portion of the material, everything that we covered after the second midterm. So that would be the second part of disinfectants lecture, antibiotics, the one that I posted last week, and today's lecture, antibiotics resistance. So the idea is that students are expected to start studying for the test. And as you go through the material, if you have any questions, something is not quite clear, well, the first thing to do would be to check the recordings. Obviously, it's all the information is there, but if you still have some concerns, whatever, you can always pose those questions next week, the last week before the exam. You can also bring your calculation questions, because calculations will still be a part of the final exam. Make sure you try the second or the third practice calculation set. It's been posted some time ago. Check it. I'll be posting the answers. I don't know, next week, I guess, before probably I kind of want to post it after the lecture so that people like later, so that people still have more time to solve those, because I doubt that most people will even start looking at the calculations, by Friday next week. But we'll see. I'll probably post it before, so you can have a chance to take a look at solutions. And if you still have questions, you can address them during the Q&A session next week. Um, so the exam, couple of things before we start about the exam. So the exam is next week, week 14, as always for all the exams, or most of the exams anyway. I think I've heard some exams are running earlier, but all of them should be on week 14 anyway. So we'll have it on week 14, and then as some of you already emailed me, and week 14 is the Friday of week 14 is a Good Friday, so um, obviously there will be no classes. There will be no the college will be closed. We won't be able to have the exam then. So that's why I posted and I emailed everybody a few days ago asking whether you have any exams on Thursday. There's probably a handful of people who will have a GNAD, the course I think it's called. So it's only a couple of people that replied so far. So to those of you who have this test for the GNAD on Thursday, I will come up with some sort of a alternative time. But as far as the rest of the students, I don't think you have any exams on Thursday. I checked with biochemistry. I checked with recombinant DNA. You don't have any exams um, on Thursdays. So we will most likely. I'm trying to finalize this with the I guess the office to book the rooms for us for Thursday next week, week 14. And it's most likely going to be in the second half of the day. So sometime around 5 or 6 p.m. around that time. Because again, like I said, Friday is unavailable, unfortunately. So we'll have to change that. So again, double check with your exam schedule. Uh, I haven't finalized the date yet, but um, it will probably happen coming Monday. So once that date is finalized, that's when we're going to have the exam. So if you have a clash with an exam in the college, let me know. You shouldn't. I'm not sure about the GNet course, how many students are actually taking that and how many students are actually having. Probably different sections might have exams on different days. But anyway, make sure you reply to that email that I sent if you happen to have a clash. If you don't happen to have a clash, no need to reply. Okay. The exam will be multiple choice, as always. Same format. I mean, it's nothing really new here. Same type of questions, calculations worth 2 marks, and all that stuff, right? Um, Only the lectures covered after the second midterm will be tested on the exam. So, like I said, second part of disinfectants, antibiotics, and antibiotics resistance. It's only 2 questions, 2.5 lectures. um, so what time on so somebody's asking what time on week 14 Thursday? Guys, like I said, check. I sent an email. I posted the message on Luminate. They should have replied by now, I guess, because I cannot wait until people, realize they have a class on Thursday on week 14, the day before the exam. We are most likely going to have it on Thursday, second half of the day. Like I said, 5:30, 6:30, maybe around that time. I haven't heard from office yet. They haven't booked a room for us. You shouldn't have any classes on that week unless it's an exam, because there are no classes on week 14. And then the other major courses that you're taking, the biochemistry and the recombinant DNA, I contacted your professors. They confirmed you don't have any tests on Thursday. So, but then again, it could be something else like GNet or something else. But to everybody listening to the lecture and everybody in the course, I posted it a few days ago. There should have been an email sent out to everybody as a copy of this announcement. If you have a time clash, you must let me know as soon as possible. If you show up and send me an email on week 14, once the days have been finalized, there's nothing I can do for you. You will have to find the time to write the test. If you happen to have a clash, check it. It takes 1 second, to get on your schedule and see if you have any. You should probably know by now what day are you having your exam on week 14 for any of the courses, or any of those courses that I might have not accounted for, like GNet or something like that. It's only a selected few people are taking that course. It's not like the whole all the people are taking it. Might be a couple, might be five. I'm not sure. I don't have the exact schedule, but in any case, check the announcements board. I posted that, and I sent an email to everybody asking to email me back only if you have a clash on Thursday at all with any of your exams. As far as the regular classes or labs, you shouldn't be having anything on week 14. Especially considering that Thursday of week 14 is going to be like the actual last day of classes because Friday is off. Alrighty, so that's that. Um, as far as the antibiotic resistance, so like I said, we're probably going to cover most of it, if maybe all of it, maybe a couple of slides will remain. It depends how fast we go, but bottom line, we can always wrap up the last few slides next week. So for the antibiotic resistance, before we start, there's a lot of information here. Everything that I covered in the lecture that was posted last week, you need to know all those details before you study the antibiotics resistance for the test. Make sure you understand the actual antibiotic mechanism of action. Okay. Don't do it the other way around. Obviously, the topics are connected. All the antibiotic resistance, as you will see today, is directly related or stems from the specific mechanism of action of antibiotics, right? Everything makes sense that way. So if you don't understand the mechanism of action of antibiotics, don't try to memorize antibiotic resistance mechanisms. You need to know antibiotic uh, mechanism of action first before you can study the resistance. If you understand the mode of action, well, antibiotic resistance is not going to be that much of a problem. It's basically a logical, I guess, continuation of what the mechanism is, right? Something about the mechanism, bacteria goes around that, changes that, and as a result, a bacteria gains resistance to that class of antibiotics. Everything is very logical and everything is very connected for these two topics. So some general considerations. There's going to be a few. I'll be adding some stuff that is not on the slides, more of a general nature, but maybe take some notes on the side to make sure you remember those things. So first of all, some basic definitions before we kick off with the rest. So what is resistance, and some of the different kinds of resistance, I guess. So pretty straightforward. Resistance is basically the ability of bacteria to survive the action of antibiotics, right? Antibiotics are supposed to kill them or inhibit them. The bacteria, remember, the antibiotics that inhibit are called bacteriostatic, static for stationary, right? Like something stationary, right? In this case, you are arresting the proliferation of bacteria with bacteriostatic antibiotics. So you arrest their proliferation, you arrest their division, and as a result, the concentration of bacteria in your tissue, in your bloodstream of the patient, or maybe on a petri dish if it's a lab experiment, bacteriostatic antibiotic stops the proliferation of bacteria, so therefore stops the increase in concentration of bacteria. It doesn't kill. Don't mix it up. A lot of confusion. Quite often people think that, well, it's because it kills bacteria, bacteriostatic antibiotic. No, bacteriostatic antibiotic doesn't kill. Bactericidal does. So bactericidal antibiotic reduces the concentration of bacteria in your site, whatever, in your location, whether it's a bloodstream, specific tissue, or a petri dish. So it drops the concentration of bacteria because it kills it. It kills the bacteria. It's not the same thing. Most antibiotics, I should have mentioned that in that video posted last week. Most antibiotics that we are using, as the ones that doctors prescribe to us, are actually bacteriostatic. Fairly common misconception. People think that antibiotics that we take are, always cidal. No, the cidal ones, there's few of them. There are some, but most of them are bacteriostatic. So antibiotic arrests the proliferation of bacteria, and then your immune system, unless it's completely overwhelmed, your immune system will take care of that whatever that infection is, whatever that infection is in your body. If your body or patient's body immune system is significantly weakened, it's some sort of a serious case, systemic infection, when the infection is in the bloodstream and is being disseminated throughout the body. In that case, the doctor may prescribe bactericidal antibiotics, so the antibiotic that will actually reduce the bacterial concentration because your immune system is unable to take care of bacteria anymore because it's overwhelmed. So as far as the resistance is concerned, ability of the bacteria to resist the action of antibiotics, right? So you throw the antibiotics at them instead of killing bacteria, or instead of arresting the proliferation of bacteria, antibiotic actually is being ineffective because bacteria is not responding to that antibiotic. Not responding in a way that its antibiotic is not exerting its effect. So obviously it is good for bacteria. They survive as a result of that, or at least some cells survive Obviously, it's not the whole population, but some cells survive, and they give rise to the progeny, to the future progeny resistant to that particular antibiotic. From our perspective, from the people, from the scientific perspective, I guess from our medicine standpoint of view, it's a bad thing. Resistance is a bad thing because that prevents us from taking care of the infection, obviously. It's a very, very fundamental property of bacteria, as you know, probably from like general type of knowledge. Um, there's a couple of important things here to understand, some misconceptions. Sometimes, especially nowadays, resistance is gaining a lot of attention in the media. People are being constantly threatened with a new pandemic, right? Well, sometimes they talk about the viral nature of this pandemic. Sometimes actually they say it could be a bacterial infection. So a lot of misconception in that regard, or some misconception, I guess, in that regard. Some of the key ones. Um, Sometimes you can hear it in the news or um, just, I don't know, maybe in newspapers or something, that people are exclusively responsible, solely responsible for the onset or for the onset of bacterial resistance. That it's our abuse of antibiotics which led to emerging resistant bacterial strains. So it's kind of it's true and not true at the same time, so to say. This kind of a statement. What is not true about it? We as a humanity, as people who are, scientists who brought new antibiotics into the market in, um, large quantities in the 20th century, we're definitely um, not responsible for antibiotic resistance as a concept. Antibiotic resistance as a concept is just as old as the antibiotics themselves, right? Bacteria, a lot of bacteria naturally lives in soil, and production of antibiotics is just one of the ways to protect for each bacterial species, individual bacterial species, to protect its own niche, right? Its own ecological niche from other species, from other bacterial species, in some cases fungi. So in order to survive in those kind of conditions, bacteria has developed resistance way before people even existed on this planet. Okay, antibiotic resistance genes, they track back all the way to way, like I said, way before the people even came onto this planet. So we're definitely not responsible for the concept itself. However, what is true about that statement is that um, mass use of antibiotics in 20th century, ever since, I guess, penicillin was purified and sold globally, right? And everything that followed after that. We obviously are responsible for the spread of antibiotic resistance, the extent of antibiotic resistance, because for very obvious, evolutionary reasons, if you try to kill bacteria with antibiotics, they will reply back with mutations and resistance as a result. We will look at some of the mechanisms in details today, so we'll cover that. And the more you use antibiotics, the more you apply antibiotics, the more you're selecting for bacterial species as well as molecular elements, molecular like plasmids and transposons, all those molecular vehicles, genetic vehicles, genetic carriers of those antibiotic resistance genes. As we will learn later today, some of these antibiotic resistance factors So those plasmids I'm talking about, transposons, and the genes, antibiotic resistance genes, ultimately, they can be actually acquired by bacteria, but they can be also lost by bacteria. They can be lost by bacteria. So if bacteria is not exposed to antibiotic, um, generally there's no point for bacteria to accumulate those antibiotic resistance genes. There's no point for them to mutate, for the bacteria to mutate its own genes and so on and so forth, right? But if you keep constantly exposing bacterial communities to high levels of antimicrobials, and it's not just antibiotics, very important to understand here. Um, I said specifically antimicrobials, right? So it's not just antibiotics, but also antiseptics and also disinfectants. If you keep constantly exposing bacterial community to these antimicrobials, you definitely are constantly promoting the evolution, right, of antimicrobial, antibiotic resistance, right? The more you use those agents, the higher is the chance the bacteria will mutate. The higher is the chance the bacteria will accumulate those plasmids. The higher is the chance that those plasmids will be passed on from one bacterial cell to another, right? So, which contributes to the spread of antibiotic resistance. Because the plasmids can be present in the cell, right? But they may not necessarily be spread actively within the bacterial community. They only are being spread if there is a need for those plasmids or for those transposons or whatever have you. So obviously, in 20th century, when all these antibiotics, as well as the rest of the antimicrobials, have been introduced, and considering how much we use them and how much we actually quite often abuse them, as in we use them without much need, that definitely contributed to all these issues we're facing, or potentially facing in the future, what you can hear in the news. As far as the extent of this threat, it's a very complicated question. There's definitely, you might have heard that back in the day in 20th century, around 1960s, uh, some WHO officials claimed that um, we defeated infectious diseases. Humanity has nothing to fear anymore, so to say. So that was said sometime in early 60s, if I'm not mistaken. Um, And now, some 60 years later or so, all of a sudden, everybody from the same WHO and other organizations are claiming that, dark ages of infectious diseases are upon us. So how is this possible? Um, so it is possible because back in 60s, there were so many antibiotics placed, uh, in the production. There were in the production so many antibiotics that um, people actually thought that we can eradicate all infectious diseases and never think about them anymore ever again. But people underestimated resistance. People underestimated resistance and the speed of, I guess, the rate of resistance spread. So now, 60 years later, after that first statement, we are facing a lot more issues than we thought we ever would. Um, a lot of factors contributed to that. I was mentioning to these kind of things before to you. Not the last factor is the excessive use of antimicrobials, the ones that are not necessarily antibiotics, but all sorts of disinfectants and all sorts of antiseptics. Not anywhere you go, but I mean, from TV commercials, from lots of different sources, you will always being pushed this idea that, you need another disinfectant in your life. You need another antiseptic to keep the house clean, to keep the house safe and whatnot, right? Those kind of messages. Um, the way the antibiotic resistance is, and in general, antimicrobial resistance is organized for bacteria, uh, it is very common that um, an antiseptic can actually lead to development of resistance, not just to that particular antiseptic, but also to antibiotics. Even though the antiseptic that we're talking about and the antibiotic are not linked to each other in any way. But the way those antibiotic and antimicrobial in general resistance genes are organized in the plasmids and the transposons, using one agent, for example, an antiseptic, can lead to selection for resistance, not just to that particular agent, but also to antibiotics that this bacteria, this particular bacterial strain, may have never even encountered in its lifetime before. So by abusing all these antimicrobials, you are definitely, we are as a, we as a society definitely contributing to spread of resistance to all sorts of antimicrobials, disinfectants, antiseptics, as well as antibiotics, and that just accelerates this process. So, and that's when you hear in the news that in the hospital there was some super resistant bug that was isolated that killed so many patients, and so on and so forth. Sometimes these bugs, super resistant bugs, go global, so to say, right? Things like MRSA, methicillin-resistant Staph aureus, was a famous example. Which takes us to the next couple of definitions here. The superbug is a layman term. It's not a scientific necessarily term, but um, the better words would be, the better terms would be MDR strains, multi-drug resistant strains, and pan-resistant strains. So make sure you know what those means. What those mean. MDR strains are specifically bacterial species of any kind, doesn't matter, gram-negative, gram-positive, which show the resistance to at least 3 different classes of antibiotics. Okay, not just 3 different antibiotics, but 3 different classes of antibiotics. Obviously, because resistance to different classes is a lot more dangerous, because if bacteria is resistant to a specific class of antibiotics, then none of the antibiotics, none of the individual representatives of antibiotics of that class will work against that bacteria, which is obviously a lot of antibiotics completely taken away now. So MDR species are the ones that show resistance to 3 different classes of antibiotics. Pan-resistant species are even worse, as the pan word suggests. It's resistance to all known antibiotics and antibiotic classes in use. Sometimes there could be some exceptions, usually in the form of specific antibiotic, one antibiotic, not even the whole class, but a particular representative. But generally, pan-resistant strains, they're obviously a lot more rare than the MDR strains. Both are obviously super dangerous. You absolutely don't want to be contracting any of these, especially pan-resistant strains. This is almost like a death sentence, because there's just no antibiotics to treat this bacteria. And if your immune system is unable to cope with it on its own, that's it. There's no solution. Unfortunately, it's not like we have a choice, right? Like when somebody contracts an infection, it's not like you have a choice of picking a regular strain or MDR strain or pan-resistant strain. Um, Most of the strains are regular strains, but MDR strains are also present in hospitals, especially in hospital settings, as we will discuss later. Uh, People can totally, absolutely contract them. And if they do, this can be a very, very serious case. Some patients never make it, and they die because of these infections. You probably heard it in the news. Probably even know somebody who contracted one of these strains. Um, so, like I said, both are very dangerous. MDR obviously a little bit less so than the pan-resistant. Pan-resistance is just very, very dangerous, almost a death sentence to a person, especially if the person is like either very young, like a toddler, or like elderly person, with both of these categories of people having weakened immune system. So obviously they have much lower chance of surviving uh, contracting this infection. Um, we'll look at some of the representatives. We'll discuss the mechanisms, but for now, just know the definitions. The definitions. Now, going back to, I guess, the discussion that I started about how we are, uh, according to all these officials anyway, how we are about to enter the dark ages, so to say, for infectious diseases. these dark ages for the infectious diseases is something that humanity lived for, centuries and centuries before the start of the 20th century. Because before then, before the sulfa drugs were discovered, before the penicillins were discovered, there was just absolutely no reliable way of treating infectious diseases apart from like folk medicine, right? All those, um, uh, you know, not very effective, obviously means very little, quite often grounded in science. For obvious reasons, that was the case because people until late 19th century didn't even know how the infectious diseases were, um, which agents, which biological entities would cause the infectious diseases, right? And we know now that it's bacteria, but before 20th century, people didn't know what it was, so you can't really treat what you don't know. So that's how the humanity lived for all of its history. So it's only basically 100 years, like literally 100 years, a little bit less. Well, a little bit more actually, because the sulfa drugs, if I'm not mistaken, they came about the 20s, 1920s. So we are basically just past a century of history of antibiotics. So why is it all of a sudden, um, it's being projected that 21st century is going to be the dark ages for infectious diseases? Don't we have a lot of drugs by now? We do and we don't. So as always with these things, there's no one clear definitive answer. So of course we have a lot of antibiotics that have been synthesized, synthetic, semi-synthetic, natural. Well, mostly these days, semi-synthetic. Natural ones are barely effective these days because they've been abused. They've been used a lot before. Bacteria has learned how to defend against it. So, for example, natural penicillins are very little used these days because there's a lot of resistance to them. So we do have a lot of antibiotics, but like I said, the key point was the underestimation. The key reason why we're facing what we're facing was the underestimation of the rate of antibiotic resistance. Um, it's not like initially people didn't know that it was such a thing, but very quickly people realized that bacteria is able to mutate, obviously, in response to these. So the rate of antibiotic resistance spread, the rate of antibiotic resistance development, and also something of a more trivial, I guess, nature, which has to do with, our society, specifically the economics. Um, Back in the day, 20s and 30s, 1920s, 1930s, when there were no antibiotics on the market, any new antibiotic was basically a success, right? Any new drug, as long as it's safe, as long as it's actually effective against bacteria, because not every antimicrobial agent is equally effective, but any drug you would find would be a potential candidate to be an antibiotic. Those new candidates were never mass produced by humanity before, so bacteria had very low levels of resistance. So all of that combined um, was the reason for low prices for antibiotics. Antibiotics were cheap to find. There were a lot of them. And the new ones, right? The new classes, the new backbones, chemical backbones for those antibiotics. So everything was very new. Everything was very cheap and affordable and effective. Um, as time obviously progressed, bacteria was getting exposed to those antibiotics on large scale. Obviously, it was a global market, so everywhere, right? Billions of people, every hospital, everybody had this some sort of antibiotic. Eventually, antimicrobials of a general nature kicked in, all those antiseptics, disinfectants, and like I said, those are contributing to the spread of antibiotic resistance as well. So with all that pressure for 100 years, people started realizing, the scientists started realizing that uh, older antibiotics don't work anymore, right? Because of resistance. So those drugs, we started losing them. They're just not effective anymore. Certain other antibiotics were found to be toxic, more or less toxic to people. Some more, So some less so, but toxic. So as we were getting new drugs, on the market, people were discontinuing certain other brands, right, or certain other types of antibiotics. But the problem is, and still remains, that bacteria doesn't really, operate in terms of economics, right? They just mutate, and there's lots of them everywhere in the soil, in the water, in the sewage water, in households. Every surface, everything around you is covered with bacteria, and all of those species are obviously capable of mutating. So for bacteria, there was absolutely no such thing as, elevated costs or anything like that. So bacteria keeps mutating. They keep finding the ways around our antibiotics, while for us, new antibiotics are becoming progressively more and more expensive to the point where it became way too expensive for pharmaceutical companies. And this topic is actually a topic worth studying on its own kind of deal. Pharmaceutical companies, don't forget, they are commercial companies. They must profit from what they make, not just because they're greedy people or something. I mean, that's probably accurate as well, but they need a lot of money to reinvest back into research, right? We spoke about it in the very first lecture. A lot of small pharmaceutical companies that came around, or not so much the pharmaceutical companies, but just like biotechnological companies that came around with the discovery of DNA and the discovery of antibiotics. They did not necessarily made it too far because they didn't have enough money to reinvest back into research. And eventually, bigger companies just obviously bought them, right? So all of this research requires a lot of money, and uh, antibiotics is no exception. It costs hundreds of millions of dollars to put a completely new, novel antibiotic on the pharmacy shelf. It's not cheap. So it's the research itself. You got to find a new backbone, and again, don't forget, all of these factors are kind of coming together for this discussion. Um, Finding a new backbone, a new molecule that will be a successful antibiotic, um, is getting progressively harder as the time passes by, because uh, a lot of readily available antibiotics, readily available for our discovery, have been already discovered. So it's not like you can just randomly, infinitely rather, come up with new backbones at will. That's not necessarily how it happens, obviously, right? Antibiotic has to have very specific properties to it. It has to be uh, effective, right? It has to kill bacteria or inhibit bacteria at sufficient levels. It has to be also safe for us. It's one of the primary considerations. So these novel backbones of novel antibiotics, especially if you're looking for something with a novel mechanism of action, like a new class of antibiotics, these antibiotics are not necessarily just infinitely, lying around, so to say, waiting for you to discover them. So this process of search for the new antibiotics is just intrinsically very expensive. We run out of these backbones. And another major contributor to this problem from a pharmaceutical company is, well, a couple of them. All the clinical trials, as you know by now, are very expensive, right? They take a lot of time. Don't forget about this parameter. We will talk about it a little bit later. Time is actually a very important thing. So it takes a lot of time to fully research and approve that antibiotic. And lastly, third or fourth, I guess here, the parameter. Um, so once you find the new antibiotic, you realize that bacteria can develop resistance to that antibiotic very quickly. Okay. And has been found in some cases for certain antibiotics that um, it takes a year or even less in some cases for the bacteria to become resistant to the novel antibiotic that the pharmaceutical company just came up with and spent hundreds of millions of dollars to find it, to research it, and to approve it, to show that it's safe and all that is safe and effective. While bacteria within the first year of commercial use of this antibiotic already shows, you already see the reports of uh, bacterial species being resistant, were found to be resistant to that particular antibiotic. Um, so, as you can imagine, this is not a very profitable and it's not a very reliable, I guess, economical model, right? When resistance develops so fast, while on our side, on our end, research takes so much, both money-wise and time-wise. To make things even worse, um, which is a very not maybe unique, but kind of unique. I guess not every drug is like that. There's one more thing about specifically antibiotics that makes it even harder for us to fight bacteria. It's a very simple thing. If you think about it, if somebody contracts an infection, most people, right, unless it is a very young audience, people with poor immune system yet, or it's the opposite end of the spectrum. You have elderly people with a weak immune system. Everybody else, if you contract an infection, unless it's something really, really bad, how long does it take you to recover from that infection? It doesn't take you long, right? Especially if you're using antibiotics, right? If, say we have a good antibiotic that is capable of treating the infection. So the antibiotic sells, it makes money, but it makes money only for so much, for so long. Because as soon as, how long does it take to cure the infection? A week, maybe a couple of weeks, depending on the severity of the infection. That's it. After that, that antibiotic for that particular individual may never be employed, may never be used for God knows, like another couple of years, I don't know, even longer, right? So uh, this is not helping pharmaceutical companies from the economical standpoint of view, obviously. Antibiotics, simply put, don't make enough money. And I did have a back in the day a discussion with um, the guy who was research and development, head of research and development of Pfizer, pharmaceutical company in North America. He was actually Canadian, but he was the head of the research and development of Pfizer. Very nice guy, very honest, surprisingly. At least it was very surprising to me back in the day. But he straight up outlined that the conversation was mostly focused on antibiotics and how we are lacking enough antibiotics, and what Pfizer, as well as other pharmaceutical companies can actually do to help with the case. So he straight up in his talk, he outlined the major 5 or 6 classes of drugs that make the most money to pharmaceutical companies, and those drugs I may not remember all 5 of them right now. So those were um, the drugs related to heart conditions, obviously. Number one, probably cause of death, right, these days. Cancer related, obviously, drugs of all sorts of drugs related to neurological disorders, right? Painkillers, number 4, or in no particular order, I'm giving it to you, just enumerating. And I can't remember what else. There was probably one more category in there. So the point being is that all of these drugs, the painkillers, the cancer drugs of all sorts, the good, so to say, quote unquote, thing about those drugs is that people suffering from the relevant conditions will likely be taking these drugs for extended periods of time, for long periods of time. Some people with um, curable, cases of cancer can be on the drugs for decades, right? And for those decades, that particular patient is paying, like either the patient or the government, whatever, but the pharmaceutical company is getting money for that drug from one particular individual for decades on a regular basis. Same applies for heart conditions. Again, there are always exceptions. People die to those conditions, so obviously, but nevertheless, there's a big portion of people who actually take them for years, these drugs. Same with neurological disorders. Same with painkillers. There's always a huge market for it. So these drugs, even though they also need to be modified and, you know, um, additional research has to be done on them, they have to be improved. But these drugs are generating enough income for pharmaceutical companies. So they keep reinvesting in those drugs and they keep getting the profits back. With antibiotics, it's like I said earlier, exactly the opposite case. Somebody contracts the infection, a week, one course of antibiotics, a week, maybe 2 weeks, that's it. The patient is cured and may never require those drugs for another God knows how long. So all of this combined, again, there's other reasons to it, but some of the major ones I gave you. All of this, as you can imagine, led to a very natural and very unfortunate, but very natural, I guess, um, slump in, as you can see on this figure, in the interest of pharmaceutical companies towards antibiotics. This figure here shows you um, how many um, new drug applications. We discussed those in the first or second lecture, right? So how many new drug applications were submitted over the past several decades to FDA? And as you can see, the numbers are dropping. They were dropping consistently. And then at some point, 2005-ish, 2009, because of all the attention to the antibiotics and the lack of the good antibiotics, pharmaceutical companies started kind of going back, reversing this trend and putting up more antibiotics on the market. But the trend is still not very favorable to us. There's not enough new drugs being made. There's not enough new classes of antibiotics being created. And again, for multiple reasons, for all these reasons that I just mentioned. In some cases, it is just impossible to come up with an effective new drug in a short period of time. So that was the trend, and still, it's not that much different these days. This timeline here on the next slide basically illustrates something that I already mentioned to you before. What you have here on the right, you have the timeline for different antibiotics when they were introduced to the market, the year of the introduction. And on the left-hand side, the year when the first reports of antibiotic resistance were that were published. So what you can see here that for some antibiotics, for example, tetracycline was a huge antibiotic back in the day. It was first introduced in 1950, and then for 9 years at least, there were no reports of tetracycline-resistant species. So 9 years is something, right? But for certain antibiotics, like for example, erythromycin, as you can see here, 1953 it was also very widely employed, and I still think it's still being employed to an extent. 1953 it was introduced. 1968 when the first reports came out. So again, significant period of time when there were no major erythromycin-resistant species out there. The point being is you don't have to memorize any specific numbers here. By the way, I'm not going to be asking you about any numbers on this figure either, but you need to know the trend. The trend is what I'm looking for, your understanding of the trend. So, but what was happening eventually, if you see at some of these values down below, the time gap, the time lag between the introduction of antibiotic and the first reports of antibiotic resistant species became much, much shorter. Okay, couple of years, sometimes even the same year. Like, for example, with levofloxacin, this guy here, the quinolone antibiotic, it was introduced in 1996, and in the same year, they already had the reports of antibiotic-resistant what is it? Pneumococcus, yeah. So same year, there's barely any time. It doesn't obviously mean that this, you know, resistant species will spread across the globe immediately. No, but nevertheless, the resistance is there, and you never know how fast it will spread. Because some of those antibiotic resistant genes, they may spread pretty quickly. They might be present in the community already. So all that has to happen is just for them to get disseminated within bacterial species, and that can bacterial communities, and that can happen pretty quickly. So the first report is always kind of like a starting point, right? And if it's so close within a year or two to the actual introduction of antibiotic, obviously that doesn't help the pharmaceutical companies to invest into this research because for them it becomes just absolutely, for all good and bad reasons, a bad investment. They don't get the profits back from these drugs. They have to come up with the new ones on a regular basis and spending more money on it, spending more money on the clinical trials and whatnot. And once they do, these drugs don't make enough profit. So obviously, this is not a very sustainable model. It doesn't matter how you look at it. I mean, like I said, pharmaceutical companies, they need to reinvest money back into research. You can't just expect them to, generate the money out of nowhere. In some cases, if you look closely, again, just for the sake of the discussion, in some cases, there were actually reports of the drug or antibiotic resistance to the drug that was identified while the drug was still being under clinical trials. Okay, so the drug is not even on the shelves yet. I mean, people already know about its existence. The scientists know that it's been that it's in the making, but the reports, antibiotic resistance reports, already there before the drug even ended up on the shelves. So that's just the worst case scenario, right? So you're working towards something that is bacteria already resistant to. So that's that. Again, don't have to memorize any specific years, but make sure you understand the trend. It's not that complicated. So all of this, like I said, it's only we're scratching the surface here. It's a very interesting field. It's a very interesting uh, topic. There's more to it, but I tried to highlight some of the most um, key, I guess, considerations in this discussion. What makes the antibiotics so hard to work on from economical standpoint of view, from the scientific standpoint of view. Another thing is to keep in mind is that antibiotics are very complex molecules, usually from the chemical structure standpoint of view. So, which means for a lot of antibiotics, we cannot really synthesize them in the lab. We still rely on natural sources. Bacteria and fungi are still better in making these antibiotics than we are, because again, the structure is still too complicated for us to replicate. So again, that also contributes to lack of new readily accessible, cheap antibiotics. So, so that's that. The scientists nowadays are looking into a lot of people in this field actually projecting that, in general, this model that we've been using for the past 100 years is not sustainable in the long term. So we have to come up with something like sooner or later it will fail. If not today, then tomorrow. If not tomorrow, the day after tomorrow, it will fail globally. Also, don't forget to factor in the globalization of the planet. People are traveling constantly, which obviously helps spread unusual infectious agents across the globe. So that is also not necessarily a factor that was present to this extent anyway. 100 years ago, people were not traveling nearly as much as they are these days. It is also a legitimate factor. So again, like I said, lots of things here. Only I try to talk about the most important ones. So to give you a more or less perspective on uh, where we are standing with this one. Don't forget the antimicrobials in general, abuse of those, massive abuse of those. People like to have, you know, protection everywhere, as they say in commercials. All those Lysol commercials we discussed, I think, briefly. Those before, people spraying Lysol all over their place. All of those things contribute to antibiotic resistance. Antimicrobial soap, absolutely no need to use it. Uh, you are not going to completely shield yourself off from bacteria. Not to mention that it's not required. You want to be exposed to reasonable amount of bacteria. Obviously, you have to wash your hands, so to say, but you don't have to kill bacteria every time you're washing your hands, because killing bacteria promotes the resistance. All that triclosan that is added to the soap will be sooner or later will end up in the soil, will end up in the sewage waters. So it will promote the resistance, will help promote the resistance. And if you think how many people, millions of people, um, are using these kind of products, to give you an idea about the scale. So obviously we're not helping the case with all these antimicrobials. So where possible, we should be avoiding using those. Obviously, there are instances like, sterilization before surgery and things like that. Obviously, there you have to sterilize and use the most harsh chemicals you can possibly find. But using antimicrobial soap in every household just to quote unquote stay protected, as they like to put it in the commercials, that's not a very smart um, approach from every perspective. And don't think that it's something that just my personal opinion or something. Um, People have been trying to influence, like scientists, major scientists in this field, were trying to influence government uh, to avoid mass production of these antimicrobial soaps and ban them from being used in households. It's not a topic for a discussion. Like, everybody in the field knows that they are no good in the long run. But unfortunately, things they do sell well, generate profit, and so on and so forth. So it's still there. But keep that in mind. Alrighty. So now let's move into some details, a little bit more details as to how the resistance works. Now that we understand what kind of problem it poses, so how exactly does it work? So this is just some major MDR species. So don't forget the MDR species. The ones that are dangerous, particularly dangerous because they are resistant to 3 different classes of antibiotics. Um, you need to know all of these for the test. It's by no means all of the MDR species out there. They're just selected few. Okay, but you need to know those. So major gram positives, MRSA. Everybody knows about that one, I think, by now. Methicillin-resistant Staph aureus. Know what it stands for, MRSA. So methicillin, we will look at it in a second, was a very important drug, still is, but until some time ago, people realized there is this Staph aureus species that is totally resistant to it. And the importance or the danger associated with this drug was so high that they even, the importance of the antibiotic was also so high that they even came up with a specific name for this bacteria, methicillin-resistant Staph aureus. It just shows you how important this drug is. VRE, vancomycin-resistant Enterococcus. Vancomycin was actually the drug that was introduced to fight MRSA, and it did so efficiently, successfully, but eventually. So as you can see, we came up, well, bacteria came up, Enterococcus specifically came up with the mechanisms which made vancomycin also not so effective. So VRE, or the vancomycin-resistant Enterococcus. Vancomycin is also highlighted. You will have to memorize this antibiotic, as we will I think we discussed it last week in that video that I posted anyway. So vancomycin is a very important drug, still a very potent drug. A lot of research is being done on it. It's one of the most researched on antibiotics out there. Penicillin-resistant streptococcus, Mycobacterium tuberculosis. You've heard about these before. Clostridium difficile. You can also highlight this one. We will be talking about it a little bit more at the end of the lecture. It's a most common hospital strain out there. Out of all the bacterial species out there, Clostridium difficile is the most common bacterial strain in the hospitals. If you ever contract Clostridium difficile and you go to your doctor, the first thing they should probably ask you whether you have been admitted to hospital recently, because that's where most people pick it up from. Major gram negatives, MDRs, just four you need to memorize. Acinetobacter baumannii. By itself, this bacteria is not the most dangerous one. I mean, there are species that are capable of causing lethal outcomes because of their resistance, absolutely. But they're also very well known for being, you can think of it as banks for resistance for other species. So Acinetobacter baumannii was found to accumulate some of the biggest plasmids with antibiotic resistance genes out there. So they just store those plasmids with massive plasmids size-wise with lots of antibiotic resistance genes. And not only they pose the risk themselves, the Acinetobacter baumannii, but it also can spread these genes to other species, right? Making it extremely dangerous. It can spread it to Pseudomonas, it can spread it to E. coli and so on and so forth. So Pseudomonas, E. coli, Salmonella, classical, your classic gram negatives. E. coli obviously can be very dangerous. Salmonella, Pseudomonas, because of their ability to form biofilms, as you know. So these four AMDR species. So again, don't make this mistake. Don't make sure you're not confused about it. It's not like every E. coli species is an MDR species. No, there's lots of absolutely harmless E. coli species. The ones that we work in the lab, absolutely fine. But within this genus, right, E. coli, you may have MDR representatives. So clinically relevant species of E. coli can be MDR. So they can cause some of the most nasty infections out there. Again, this is not a complete list. You don't need a complete list, but just some of the major um, gram negatives and gram positives that are capable of being MDR species. So that's that. Let's make a break, I guess. It's been an hour. It's been a lot of information. So let's make a break, come back in 7 minutes, and then we will start talking about specific mechanisms of resistance. We'll talk about what kind of organelles are affected and so on and so forth. So let's make a 7 minutes break. All right, let's get back to the lecture. So the major mechanisms of resistance. We will look at first, I guess, general classes of resistance, right? Um, and then we will look at specific classes of antibiotics and how those classes of antibiotics develop resistance. So these classes here is a good way to, you know, categorize, I guess, all the resistances based on how those resistances are acting. What's the mechanism behind them? So that will help you kind of get a big picture as to what's happening on a molecular level. So the four major classes are enzymatic inactivation of the drug, alteration of the drug target, reduced cellular uptake, and increased efflux of the drug. So most of the antibiotic resistances can be classified into these four categories. One thing to keep in mind, to make a note of, these mechanisms of resistance, these classes of resistance, or groups of resistance, are actually arranged in the order of decreasing clinical importance. Decreasing clinical importance. So enzymatic inactivation of the drug being the most clinically relevant group, and increased efflux is being the least relevant, clinically relevant group. So what do I mean by clinical relevance? A lot of you probably don't know what it means. So basically, when you look at these four groups, the question that scientists posed back in the day, or they just looked at all different mechanisms of resistance and they decided to organize them in terms of which of these classes, which of these groups gives the most clinical trouble to us, which gives us the most trouble in clinical settings. That's what clinical relevance is. So that means bacteria that show enzymatic inactivation of the drug, that have the antibiotic resistances that fall into this category, are the ones that give us the most trouble in clinics. That means a drug, bacteria with this resistance is usually uh, causing severe infections that are hard to treat because of resistances. And then bacteria that is showing resistance due to increased efflux of the drug, showing the least clinical relevance, meaning it is the least rather represented amongst the bacterial species that are causing severe infections. It doesn't mean that they just don't exist. It doesn't mean that bacteria that show increased efflux of the drug are not capable of killing people. They are, but again, compared to the rest of the classes, this is the most rare, the least represented one, while enzymatic inactivation being the most represented one. Uh, this, in its case, in its turn, rather also gives you an insight into how effective, right, from clinical standpoint of view, how effective these mechanisms are. So enzymatic inactivation being that clinically relevant is one of the most effective ways for the bacteria to generate resistance. It's the most common and the most effective, and that's why, as a result of all of these, it gives us so much trouble in clinics. So that's that. Make sure you remember this relationship, or this trend rather. So from the most clinically relevant to the least clinically relevant. So let's look at these four groups. Just make sure I understand what's hiding behind what are the actual mechanisms behind each of them. It's nothing really complicated about it. The last maybe two groups would be new conceptually to a lot of you. So we'll discuss those in details as well. So the first one is enzymatic inactivation of the drug, just as the name suggests. Again, a lot of this information can be, uh, you know, can be figured out by just understanding the title, right? Understanding the name. So enzymatic inactivation of the drug. So what do you think bacteria does that exhibits this resistance, right? It enzymatically inactivates the drug that you throw at them. So you throw the antibiotic at them, they secrete the enzymes, the proteins, the enzymes, which will degrade, that can degrade your antibiotic. The very famous representative, the one that you've employed even in the labs, in BI 312 labs, unless they change something about those labs, is the uh, beta lactamases, the enzymes, beta lactamases. And you were using it, you were making use of this in the lab where you were uh, expressing the P-glow plasmid. So the P-glow plasmid on the in your cells was supplied with ampicillin resistance gene on it, remember? And then you had to add antibiotic to the media so that the bacteria that has been successfully transformed with this antibiotic with this plasmid also gained the resistance to ampicillin because of the presence of that gene. So that gene, ampicillin resistant gene, ampicillin, as you should remember from last lecture, is a beta-lactam antibiotic. Okay, and the gene that was present on that P-glow plasmid was the one encoding for beta-lactamase enzyme that would cleave ampicillin drug once that drug was in the media. So the drug was being cleaved and bacteria was able to survive. So ampicillin resistance is mostly due to beta lactamases. The enzymes, as the last part of the word, ase, refers to the fact that it's an enzyme. So it's the enzyme that cleaves beta lactam antibiotics. Make sure you understand what it means. So this is just one representative. In general, this is a group where you have antibiotic inactivating enzymes. Okay, so the chemical structure is being cleaved, and therefore antibiotic loses its ability to do its job. The next group, alteration of the drug target. So basically a different mechanism achieves similar results, I guess. Is intended to protect bacteria from the antibiotics, but in this case, for this category, it does it differently. Bacteria doesn't secrete any enzymes that would cleave the antibiotics, but what they do instead, they protect their own organelles, the ones that are supposed to be acted on by the antibiotics, right? Which is very obvious. Always think of analogies, make sure that kind of settles in your mind, all these mechanisms settle in your mind properly. It's like, you using a key to open the door, right? So the key being your antibiotic, so to say, right? The door lock is being your bacterial or organelle, some sort of organelle in the bacteria, a ribosome or the cell membrane or cell wall. So one way to protect, for the bacteria to protect itself, in our analogy, one way to not have the interaction between the key and the door is to do something to the key, right? You break the key. So that would be like an enzymatic inactivation of the drug. The other approach would be, I don't know, to like stick something into the door lock so that the key cannot go in. So that's pretty much what this mechanism is about. And certain mechanisms, specific mechanisms that we will talk about, specifically ribosome protection, is actually conceptually is not that different from what I just told you about the key and the lock. Bacteria literally protects its ribosome in a very funny way. We'll talk about it later. And after that protection, the antibiotics cannot attach to ribosome anymore. And if they cannot attach, they cannot change the expression of the proteins. So again, for the second group here, antibiotic remains untouched by bacteria. What you do instead, what bacteria does instead, it changes, it protects one way or the other the target for that antibiotic, its own organelle, whether it be ribosome or whether it be some sort of a penicillin binding proteins. We'll look at those in a second. It protects its own organelle, its own sometimes enzymes, the ones that are supposed to be acted on by the antibiotic. The last two that probably are very new to most of you, the reduced cellular uptake and increased efflux of the drug. So what are those about? They're actually pretty neat. I will have, can't remember if it's the first, no, it's probably, oh, sorry. No. We'll have I'll have a nice animation on this in the next few slides. So again, as the name suggests, always start from the title, from the name of the term, reduced cellular uptake. So this has something to do with the cells, uptaking less. So what do you think is happening in this case? Bacteria protects itself by shutting down, essentially, the doors for the antibiotic to come in, right? So they just change something about their cell structure. We'll talk about it later. So some alterations in the cell membrane happen, and because of those changes, antibiotic loses its ability to enter into the cell. And if it cannot enter into the cell, it cannot, most of the time, it cannot get to the organelles, the ribosomes, whatever have you, DNA. And if it cannot, it doesn't have access to those structures, to those enzymes, or to those molecules. Obviously, antibiotic is unable to uh, exert its effect, right? So you just basically, like I said, bacteria is basically like shutting down the doors on the antibiotic. Now, increased efflux of the drug is very similar, conceptually related to the reduced cellular uptake. So, and a very visual mechanism too. If antibiotic still inevitably ends up inside of the cell, which most of the times will be the case. So if antibiotic leaks inside of the bacterial cell, bacteria has this fancy mechanism where it will literally pump the antibiotic out from inside of the cell into the outside of the cell, into the um, extracellular, into the space outside of into the space outside of the cell, right? It literally pumps it out just like a pump would pump out water from the basement or something. It even the way it is structured, those efflux pumps, they kind of look like pumps. They physically operate like pumps in a way, molecular pumps. We will look at some of the animations for those. So what they do in this case, they do essentially the same thing as reduced cellular uptake, which is in both cases, bacteria significantly reduces the effective concentration of the antibiotic inside of the cell. But it can achieve that with two different means. The first one is where you prevent the antibiotic from going in. The second one here, the increased efflux, is when the antibiotic is already in, you just pump it out, you spit it out, basically. But in both cases, you just reduce the concentration inside of the cell. Very, very intuitive, I think. We will look at some of the animations, so I'm just introducing these to you. It will become even more clear. As far as the origins, very briefly about the origins of resistance. We already spoke about those quite a bit. Just know the basic genetic vehicles, the molecular elements that are responsible for the propagation of antibiotic resistance genes. They're being primarily plasmids, especially the conjugative plasmids, the ones that are capable of replicating itself and passing themselves to the cells next to them, so to say, right? We'll talk about what the horizontal gene transfer is later. So don't want to overwhelm you right now with this. So what are the major origins, plasmids, they sometimes are referred to as R factors. R stands for resistance. So this is just one of those layman terms for, well, it's not necessarily a layman term, but just one of the terms for the conjugative plasmids. R factor, resistance factor. Why? Because plasmids are, in the bacterial context, are usually transferring resistance, carrying resistance genes with them. That's why they're sometimes being referred to, especially in clinical literature, as resistance factors. Insertion sequences as well as transposons, those are the jumping genes. Those are the sequences that are capable of moving around in the genome. As you know, they can move um, the genes, antibiotic resistance genes, obviously in our case, from genome into the plasmid, from genome into the genome, and from plasmid into the genome. So they can exchange, they can help the bacteria exchange the genetic information, specific sequences. So you've heard about those before, I'm sure, the insertion sequences as well as the transposons. Insertion sequences can not only move the genes, but they can also move regulatory elements. And then because of those insertions in the genome, they can activate antibiotic resistance genes that were previously silent. Okay, so the gene was there, but it was silent. It wasn't being expressed. And the insertion sequence, if it gets inserted in the right way, so to say, luckily for bacteria, not so lucky for us, it can actually activate previously silent antibiotic resistance genes. So that is obviously one of the mechanisms. Transposons, again, mobile genetic elements, jumping genes as they call them. They travel randomly, they make a copy of themselves and they move into another location. Sometimes they excise themselves from the location and then they insert themselves somewhere else in the genome, hence the name, jumping genes. So those genes can move, like I said, from genome into the plasmid, from plasmid into the genome. So responsible for the exchange of genetic information this way. Recombination, obviously, recombination of foreign DNA into the chromosome, into genomic DNA, it can be recombined, and as it ends up in the genomic DNA, it now becomes a part, right? It becomes a part of bacterial genome. It will be passed to all the subsequent generations. Mutations, don't forget those, the basic mechanism for generating, genetic diversity. Mutations, all sorts of mutations. We're not going to discuss those insertions, deletions, and whatnot. So all sorts of mutations from all sorts of different physical factors, chemical physical factors. And in general, a lot of these genes, antibiotic resistance genes, are common in soil bacteria because soil bacteria is responsible for most of the antibiotic production. We discussed it last week, or in that video, I told you that soil bacteria are responsible for about 70% of all the antibiotics that we are currently using, specifically streptomyces species. So that soil bacteria is basically a natural reservoir for antibiotic resistance genes that were present there since the dawn of ages, so to say, since the dawn of time, since antibiotics, since bacteria actually started producing antibiotics way before it predated our existence. So these genes can be also moved around as part of the transposons, as part of the plasmids. They can be moved around from soil species onto clinical isolates. And once those genes end up in the clinical isolates, they are getting exposed to antibiotics and all sorts of antimicrobials on a regular basis, a lot. And obviously, this kind of phenotype will get propagated only will get further propagated from one species to another, from one bacteria to another, because of the pressures provided by all the antimicrobials, right? So this natural reservoir, like I said, is a significant source of antibiotic resistance genes for all the clinical isolates. As far as specifics of these factors, we're not gonna go too much into this. I used to have more information about this, but whatever, we don't need that much. For plasmids, just remember, conjugative plasmids are especially nasty, so to say, in this regard, because they are the ones that are capable of replicating themselves and moving themselves from one cell to another. So that's one of the main mechanisms of genetic exchange of the antibiotic resistance genes between bacteria. Again, the R factor stands for resistance factor. Don't get confused. It's not like plasmids are only known for conferring resistance to bacteria. No, it's just in the context of antibiotic discussion, antibiotic resistance discussion. People working with these like to call conjugative plasmids carrying the antibiotic resistance genes as resistance factors. Obviously, plasmids can carry a lot more than just antibiotic resistance genes. Couple of things, general things about the plasmids. I'm not asking you to memorize these specific representatives. These are just the names of the plasmids. A couple of things about the plasmids. They're very mobile, so very good from bacterial standpoint of view, moving the antibiotic resistance genes around. The plasmids can store multiple antibiotic resistance genes on one plasmid, which is again very nasty feature because if you have several genes encoding for different resistance, for different types of resistance, if those genes are under control of the same promoter, exposure of such bacteria to one of those antibiotics will activate all of the genes under the control of the same promoter. Okay, so if you have, for example, 3 antibiotics, ampicillin, I don't know, vancomycin, and methicillin, under the control of the same promoter, if that bacteria gets exposed to ampicillin only, this will activate the whole the gene cassette. All three genes will get activated, even though bacteria was only exposed to one antibiotic, just ampicillin, but it will be expressing also ampicillin resistance factors, those enzymes, vancomycin resistance factors, and methicillin resistance factors. So that's one of the mechanisms why bacteria can sometimes show absolutely unexpected resistance profiles, even though it was never being exposed to that particular antibiotic, because the genes can be organized in these kind of conglomerates, in those gene cassettes. And in this case, one antibiotic or one antimicrobial can cause the resistance to multiple antibiotics. So just remember that. So very easily, like I said, they're very mobile, so very easily shared, very easily moved around by bacteria, so very, very potent factor. Now, let's talk specifically about efflux pumps as well as about the reduced cellular uptake, because these are probably new mechanisms to you. So like I said, I have a nice animation here, so it will make things very clear. So what are the efflux pumps? So let's actually, you know what, let's just look at the animation and I'll explain everything by looking at it. So this is your schematic for a gram-negative cell. You have your 2 layers of the phospholipid bilayer, outer one, inner one. You have your extracellular space here outside. You have your periplasm, and then you have your cytosol. So the cytosol is where all the organelles are. This is where your ribosomes, your DNA, bacterial, not your, but bacterial, obviously, elements are. And what we have here, these are actual models. They're not just pictures or drawings. These 2 proteins are actual protein models. So this guy here, the yellow one, is the protein called porine, P-O-R-I-N, porine, from the word pore. So this is basically a small protein which is responsible for the uptake of the small molecules, including water, into the cell, fairly small molecules into the cell, including antibiotics. Not all of them, but some of them, depending on the size. So this is what I called earlier, basically the doors, right? The gates into the cell. Through these proteins, the antibiotics will get into the cell. This guy here, this protein here, it's actually, as you can see, made of the 3 distinct proteins. They're color-coded. The green one, the red one, and then the tile one, tile color, light blue. So this is actually a model for one of the efflux pumps that were found in gram-negative bacteria. So this is literally works like a pump. And if you were to look at this protein from the top, we are looking at it from the side. But if you were to look at this protein from the top, you could see that it's actually hollow inside. It's literally like a pump. And when the antibiotic is moving through this pump, the pump is actually changing its shape so that the antibiotic is being pushed outward, just like, kind of like a rubber pump would push out water. Very similar mechanism. Obviously, similar from the way it looks, in the way it looks, but obviously this one is a lot more tricky molecular mechanism than just your regular pump. But like I said, the principle, the idea, the mechanism behind it is very similar. So what happens when you have such a bacterial cell with these two elements in place? So you add your antibiotic at some point to your you prescribe it to a person who has this bacteria, or you're doing this as a part of research or something. So these molecules are your is your antibiotic. So antibiotic will get inside of the cell through the pores, through the porine proteins. Porine. It will go inside of the periplasm as well as into the cytosol. Obviously, from the bacterial standpoint of view, this is a very bad scenario. This is what can kill the cell. So, how would bacteria, using these two methods, these two mechanisms, I'm referring to these two methods, reduced cellular uptake and increased efflux of the drug, how would it deal with this situation, with this scenario? So the first way to deal with this, which is referring to that mechanism here, reduced cellular uptake. The simple approach would be to preclude antibiotic from getting inside of the cell, right? You just have to stop it so that it doesn't get in there. So this is being achieved, or can be achieved. It's not always being achieved, but can be achieved by some species by a very simple mechanism. What's the very logical one? And try to memorize it this way. This way you will never forget how it works. So if you want to reduce the concentration of antibiotic in the cell, what would you do? You would shut down all these doors, right? Through which antibiotic is coming in. So in molecular terms, it means bacteria can downregulate the expression of the porins. So in other words, porins levels in the outer membrane of the cell are being downregulated. They're being produced, they're being expressed less. So fewer porins means fewer gates for the antibiotic to get into the cell. So that means lower concentration of antibiotic on average inside of the cell. So again, down regulation of expression of porine proteins. That's that third mechanism. Reduced cellular uptake. Alteration in cell membrane usually involves downregulation of the porine expression. Now on to the fourth mechanism, efflux. So let's say for whatever reason, the bacterial cell failed to downregulate the porine expression significantly, and antibiotic ended up inside of the cell. So what would be that efflux pump mechanism? So literally this efflux pump recognizes these toxic compounds, antibiotics, and it activates a whole tricky mechanism. We're not going to go into this, but it will actually bind the antibiotics both in periplasmic space as well as in the cytosol. Will move those antibiotics inside of it because, like I said, this pump is hollow inside, it's empty inside, and it will efflux them out. It will efflux them out and it will spit them back into the extracellular space. What happens in this case, as a result of this, you have a severely reduced concentration of antibiotic inside of the cytosol. Okay, which from bacterial standpoint of view is very, very good. We have a question. So, is this mechanism, so Gurley is asking, is that mechanism applied to any type of antibacterial agent as well? So, very good question, actually. I'll address it pretty much right away. So, as far as this pump is concerned, this particular one, first of all, I guess what you need to remember is that this particular pump is just one of the pumps, one of the type of the pumps that bacteria makes use of. There are also other ones in gram negatives and gram positives. This one is specifically common in gram negatives. It's literally a pretty big pump because it has to position itself exactly like this in the outer layer of the cell membrane as well as in the inner layer of the cell membrane. It has to span both layers, obviously, for it to work. But there are also smaller pumps for gram positives. There's different kind of pumps for gram negatives as well, but that's the general principle for all of them, with obviously some structural differences given, but overall mechanism is like that. Now, as far as the profile, so what is Gurley asking? As far as the profile is concerned, it's actually a very curious thing about these pumps. For the longest time, I'm not sure where the research on this topic is these days, but not so long ago, people were actually puzzled. Researchers were puzzled by these pumps. So it turns out that um, these pumps have a very, very, very wide range of substrate profile. And that's very unusual. And like I said, I'm not sure about 2025, but some 10 years ago, people didn't have the explanation for it. So this pump turns out can recognize a super wide range of chemicals, starting with small molecules, super small molecules like metal ions, all the way to bulky antibiotics, like bulky, fancy antibiotic structures. This kind of pump can recognize a very wide substrate range. So why was it unusual to scientists? Because usually this kind of interaction that happens, this kind of recognition of the pump, of the agent, of the substrate by the pump, is based on structural affinity. So it is very uncommon to see that the pump can recognize something as small as a metal ion, as well as something as bulky as an antibiotic, because certain antibiotics, a lot of the antibiotics, and certain ones especially, are really big molecules, super big molecules, as we will talk about a little bit more in the next few slides. So to answer that question, yes, this mechanism applies to, I don't want to say all of the chemicals out there, but to a lot of different chemicals out there. Also to keep in mind, for you to keep in mind, is that these efflux pumps, they serve, apart from effluxing the antibiotics, they are present in bacterial cell wall basically as a waste management molecule. They're not specifically designed by nature to efflux antibiotics, not specifically for that purpose. They are responsible for the efflux of all sorts of byproducts that the bacterial cell generates, even if that bacterial cell is never exposed to antibiotics in its lifetime. These kind of efflux pumps will still be present because they are just, like I said, like waste management, like people picking up garbage outside, right? So that's what these pumps are doing. When the cell generates all sorts of byproducts, all sorts of toxic byproducts in its biochemical pathways, these pumps are responsible for taking that garbage out. And like I said, surprisingly so for scientists, it has been found that these efflux pumps can also recognize a lot of antibiotics, a lot of different classes of antibiotics. So they have a very, very wide substrate range. Now, one thing, we kind of mentioned this briefly before, but one very important thing about this mechanism of protection by bacteria of itself. As we looked at it earlier, don't forget these are the bottom two mechanisms, right? They're the least clinically relevant. So why is that the case? Because the effectiveness of this method of protection literally depends on how many efflux pumps are present in the cell wall and how many porins are expressed in the cell membrane. The fewer porins a bacterial cell has, the better for bacterial cell. The more efflux pumps it has, the better, because obviously it can efflux more and faster. Okay. So background levels of porins and efflux pumps are always present in all bacterial cells, right? Like I said, porins are not just responsible for the antibiotic uptake, they're also responsible for water uptake. So they will be present there regardless of bacteria being exposed to antibiotic or not. Same with the efflux pumps. They're not just specifically designed for antibiotics. They are waste management of the cell. They're taking out all the byproducts. But at the base level of expression, right, at a standard level of expression of these two proteins, the concentration of porins and concentration of efflux pumps is not sufficient for this mechanism to be very effective, to be clinically relevant. Okay, so in other words, when these two proteins are being expressed at their base levels, it's still too many porins in the bacterial cell for the antibiotic to go in. And it is still too few efflux pumps for the antibiotic to get pumped out. So at the base levels of expression, this mechanism is not very effective for bacteria. Some antibiotic will get effluxed out, but not a lot. And that's not going to be enough for it to save bacteria. So when does this mechanism actually kick in, and when does it actually work, and when does it actually become clinically relevant? Only when you have significantly downregulated levels of porins and significantly upregulated levels of efflux pumps. So in other words, bacterial cell either severely decreases the level of expression of porins or severely upregulates the level of expression of efflux pumps. In either or both, they can work in concert. Simultaneous downregulation of porins and upregulation of efflux pumps, so that as little of antibiotic goes in and as much as possible of antibiotic is being pumped out. So only then this mechanism is working efficiently for bacterial standpoint of view. Okay, we have another question. How can the number of porins be reduced? So bacteria switches on certain mechanisms. This regulation happens on a genetic level. So bacteria downregulates the expression of porins on the genetic level. We can use certain agents that can block the expression of the porins. Or we can use certain inhibitors that will block the activation of efflux pumps, but it's not necessarily a very reliable method. So it mostly depends on intrinsic levels of expression of these proteins in the bacterial cell. So if the bacterial cell has a mutation, for example, in the promoters for these elements, then it might be extremely challenging to counter that. If the levels of efflux pumps are really high because of that mutation, if the levels of expression are constantly being high, it's very hard to. But certain agents being employed, yes, in clinics to specifically battle bacteria that is resistant and therefore clinically relevant because of the severe levels of upregulation of efflux pumps. These chemicals exist, but again, it's not always a reliable method. So, but yeah, so there was a previous one, I think, question that I wanted to. Oh no, that was the mechanism. Yeah. Any type of, yeah, I don't want to say any type of antimicrobial agent, but most of them, a very wide substrate range. That's the beauty of this efflux pump from the bacterial standpoint of view. Not so much for us, obviously, because we suffer from those kind of things if they happen to be upregulated. But from the bacterial standpoint of view, it's a very wide substrate range. So again, so only when this happens, when porines are downregulated and efflux pumps are upregulated, this becomes clinically relevant. It doesn't happen often, fortunately, but cases like that do exist. Absolutely do exist. There were lots of lethal outcomes due to patients, people dying because of contracting um, bacterial species infection that was uh, expressing high levels of efflux pumps. The most famous um, historical, I guess, precedent, that's how this actually mechanism in general gained, um, you know, media attention for the first time. That actually happened in 2000, what, one and two, right after 9/11 in states, right when those um, trade center was brought down by the airplanes. And what happened was states invaded Iraq, right? Afghanistan and Iraq eventually. And the first cases of soldiers coming back with really severe wounds and contracting, like the severe wounds, obviously on the battlefield, and then contracting Acinetobacter baumannii, this specific species, which were also very expressing very high levels of efflux pumps. And some of those patients, quite a few of those patients actually died because doctors couldn't do anything. So that's how this mechanism and that's how um, this general mechanism of resistance gained, uh, severe public attention. That was in 2001, 2002. It gained a lot of attention. It gained a lot of research. And to wrap up this particular topic, um, it gained even more attention and more interest when people started studying these, and they realized that, which is also a fairly recent discovery, that resistance of certain cancerous cells to chemotherapy is actually due to a mechanism very similar to this one. So, in other words, cancerous cells can express, not all of them, again, there's always, it's not a blanket statement, but there were found a lot of cases where cancerous cells express efflux pumps of a different kind, not necessarily this particular one. This is a very specific model, but of a different kind, but the same idea. And you throw all those chemo agents at the cells, and the chemo agents end up inside of the cell, but then they're getting effluxed out by the pumps. So that was also a very big thing. This topic gained a lot of attention in the media, just in general in scientific circles. So these pumps are really, really versatile as far as their substrates are concerned, as far as their host organisms are concerned, and host cells. So it's a big topic, far from being fully studied at this point. Just one of the mechanisms how the cells can defend themselves. Alrighty, so that's that. That's as much as you need to know. Obviously, there's a lot more to it, but we're gonna sufficient good enough level of details for you to understand that method. A couple more things about before we talk about enzymatic inactivation of the drug, and then protection of the cellular targets, there's one more, um, I guess, piece of information about origins of resistance that I wanted to uh, draw your attention to. All of these methods of resistance that we looked at, right? The four. We haven't looked at the first two yet. We will. But all of these methods of resistance can be classified into these categories based on their mechanism of action, but also they can be classified into something like this, into the nature of that resistance, and specifically whether the resistance is considered to be innate or whether it's considered to be phenotypic or acquired. A synonym to phenotypic resistance is acquired resistance, something that the cell acquired over time. So what is that about? This is a general, it's not even a classification. I don't want to say that it's a classification, but in a way, right? When you split it into two groups, you kind of classify it. It's a classification of resistance based on its origins. Let's put it that way, based on its origins. So what is it about? It's not very complicated. So as the title suggests, as the name suggests, innate resistance is something that the cell is born with. Okay, something that the cell is born with, it cannot lose it, and most of the time it cannot pass it to the cells of the same generation. I'll come back to this in a second. Phenotypic resistance or acquired resistance is something that the cell acquires over time, over its lifetime. And this is something that is not necessarily being passed to the next generation. It can be lost, though. Okay. So I think you're probably getting, you probably know what I'm getting at. All of the we just discussed can be classified into these two groups. So phenotypic resistance, let's start with this one, acquired resistance, something that is gained over the lifetime and can be lost over the bacterial cell lifetime. So what do you think falls into this category? All sorts of these conjugative plasmids, insertion sequences, can be classified as innate resistance, as acquired resistance, phenotypic resistance, because this resistance comes on a plasmid. This plasmid is not necessarily something that the cell is born with. It is something that it acquires as a result of adaptation to growth in the media that has antibiotic. So, for example, in that, going back to that BI312 experiment that you did, when you were transforming the cells with the plasmid, with the P-glow plasmid, and you were supplying antibiotic into the media. So the cells originally didn't have any plasmid, right? So they were born without the plasmid, so to say. But what you did, you exposed them to antibiotics, and you also exposed them to a plasmid. And you basically are telling the cells, grab the plasmid or you will die, right? So the cell is acquiring a new property, which is the antibiotic resistance, during its lifetime. It wasn't born with that property, but it acquired during the lifetime as a result of adaptation, bacterial cell adaptation to growth and survival, right? Within a very specific environment. And that specific environment was the antibiotic containing environment. In your case, it was ampicillin. So that's where all those plasmids, resistance conferred by plasmids, falls into as a phenotypic resistance. Insertion sequences as well as the transposomes can also be a part of phenotypic resistance when those genes have been moved around as a result of growth of in a specific environment. So what is this? What is the innate resistance? Let's wrap up with this one. Innate resistance is resistance due to genes, antibiotic resistance genes, that the cell is born with. So what kind of genes is the cell born with? The genes that are present on its genomic DNA, right? And those kind of genes are the genes that are usually responsible for reduced cellular uptake and increased efflux. These genes are usually, not always, but usually come from the genomic DNA. Enzymatic inactivation of the drug, alteration of the drug target, there is no real way to like place these two categories into into specific group here. Those two first ones can be either innate or phenotypic. There's no hard rule here, but increased cellular uptake and reduced cellular uptake and increased efflux, those are usually coming from the genomic DNA, and therefore they're a part of innate resistance. And as it says here, these are usually associated with variations in the structure of the cell envelope. So in other words, we're talking about these two elements, the number of porins and the number and the activity of the efflux pumps. So what are some major consequences from these two modes of resistance? Not modes of resistance, but from these two different types of resistance, innate versus acquired resistance. Acquired resistance coming from the plasmids can be gained and can be lost within the same generation of cells. That is a good thing for us and a bad thing for us at the same time. Same for bacteria. It's a good thing. They can acquire it and protect themselves against antibiotic, but they can also lose the plasmid. Why is it good for us? Why is it bad for us? It's bad for us because the cell can actually gain resistance from the plasmid, right? It's always bad. The cells are usually obviously contained, usually present everywhere around us. So if one cell has the plasmid, there's a good chance that if that bacterial community is being exposed to antibiotics, all of the cells in the community will share the plasmid. So that's bad for us. Why is it good for us? Because if the plasmid was obtained by this bacterial cells, it can be also lost. So what does that mean? That means we can reverse the antibiotic resistance if that antibiotic resistance was a phenotypic one. Okay, so what does it mean practically? It means if you have a bacterial community exposed to some sort of antimicrobial or antibiotic specifically, and because of that exposure, because of that adaptation, right, to growth in the antibiotic containing environment, if as a result of that, bacteria has acquired plasmids, if you now take that community and you remove that antibiotic or that antimicrobial from that environment over several generations, bacteria will lose the plasmid. How will it lose it? It will just stop replicating it. Because keeping the plasmid inside of a bacterial cell is actually not necessarily a cheap entertainment, so to say, for bacteria. Keeping the plasmid costs a lot of energy, and basically, takes a lot of maintenance to replicate the plasmid. Takes a lot of energy to fix the plasmid, to move it around, and all that stuff. It takes a lot of energy. So unless bacteria absolutely must have it, it is very willingly will lose it. It will stop replicating it. So from our perspective, like I said, it's a very good thing. That means there's a hope if we have a bacterial community that has acquired resistance through the plasmids, if we stop, exposing that environment to antimicrobial over a couple of generations, a couple of iterations of cell replication, we can actually see, and that has been proven obviously in experiments, we can see that the same bacterial community will now lose the plasmid. Very, very, very few cells will actually keep the plasmid, but most of the cells in that community will lose it. And what does that mean? That means we have reversed the resistance. It was there, but then bacterial cells lost it. So that's good for us. Okay. With innate resistance, along the same logic, but slightly different conclusions. Innate resistance is due to something that the cell is born with, some sort of genes that are on genomic DNA. So what does that mean from the I guess, in us versus infection standpoint of view. From bacterial standpoint of view, it's also a good thing and a bad thing at the same time, just like it is for us. Why is it a good thing for bacteria? That means this is a good thing for bacteria because if the cell happens to have that resistance gene in its genomic DNA, and if that gene is upregulated significantly because of the promoter or whatever, there's a fairly low chance, very low chance that that gene is going to get lost, and a fairly low chance that that gene is going to get downregulated. So it means that most of the time, that gene is going to be always be there for that bacteria, and it will always be expressed at whatever level it is expressed. Why is it bad? It is bad for bacteria if bacteria doesn't have the gene, antibiotic resistance gene in its genome. The chances of it acquiring that gene into the genome, into the genomic DNA, is very low. It can happen because of the transposons and whatnot, insertion sequences, like we discussed earlier, but the chances are pretty low. So if it doesn't have it, it will not be able to survive unless it actually picks up a plasmid from somewhere. But if the plasmid is not there in the community, that's it. Bacteria has no options. So along the same logic, obviously, the same applies to us. Why is it bad for us? It is bad for us that because if the bacteria has the gene, there's nothing we can do because you cannot really force the bacteria to get rid of that gene. You cannot reverse the innate resistance. Why is it good for us? Because if it doesn't have it, then it doesn't have it. The bacteria is susceptible to whatever antibiotics. So it's a double-edged sword for both us and the bacteria. Um, so that's that. One last thing is because which is direct consequence of what we just discussed, innate resistance coming from the genes that are being passed on from generation to generation, right, from the mother cell to the daughter cell to the next cell and so on. This kind of resistance and passing of the resistance genes like this is commonly referred to in literature as vertical gene transfer. Vertical gene transfer. Why vertical? Is because you're passing from mother cell to daughter cell and so on, so vertically from generation to generation. Phenotypic resistance, when the plasmids is being shared between community of cells, this is what they refer to as horizontal gene transfer. We had that term earlier. Horizontal meaning that you are moving the genes within the cells of the same generation, right? Because the plasmid is being given to the cells of the same generation. It's not being necessarily passed on to the next generation, or it can get lost eventually, but within the same generation, it can be passed on aggressively, passed on to the neighboring cells, so to say, aggressively, as long as the environment is forcing that. So that's what horizontal gene transfer and vertical gene transfer. Make sure you just understand the logic behind it. Just memorizing it blindly can be challenging. So try to have that nice structure in your head as to what's happening, why is it happening, where in the inside of the cell this information is being stored, genome versus plasmid. If it's a plasmid, what does it mean? It means that it can be moved around. It means that it can be lost, right? All those things, they should follow logically from your understanding of the topic. So that's that. Some general considerations about these things. So, like I said, both means both. All these different means of antibiotic resistance, they all have their own pros and cons, right? For us as well as for bacteria, pros and cons. There's no, you know, uh, there's very nasty cases when bacteria can have both. So it has I guess, versatility that way, and those species are usually extremely dangerous in clinics. But there's always some pros and cons for each method of resistance, for each type of resistance. The way it is encoded, the way it is actually being executed, whether it's efflux or whether it's something else. Alrighty, so would have been another hour, so let's make another break and come back in what 7 minutes at 9:49 or 9:50. We'll probably cover another five, six slides, and then the remaining four or five we will leave to next week. It won't take too long. Okay, so let's make a break, another 7 minutes, and come back at 9:50. All right, let's get back to the lecture, last half an hour or so. So what we're going to do now is we're going to look at specific the same classes we looked classes of antibiotics that we looked at last lecture, the beta-lactams, so on. And we will look at what kind of antibiotic resistance is most common for those classes. So starting with the beta-lactams, I'm not going to remind you the mechanism of action. I'm expecting to not waste time. I'm expecting people know that, or at least you should know that when you study this lecture for the test. So again, this is your general formula for the beta-lactam. You do not need to know it. It's just so that it's easier to explain what happens. So with the beta-lactams being very important clinically very important antibiotics, there are two major mechanisms of resistance that inactivate these antibiotics. The first one is the enzymatic inactivation, and the second one is the alteration of the drug target. I'll explain later what the PBPs are. So enzymatic inactivation of the drug, as the name suggests, it's bacteria secreting the enzymes, the one that will cleave the chemical structure of the antibiotic. This ring here that I'm highlighting is the ring on the left is the beta-lactam ring. So bacteria can secrete enzymes, and they're called beta-lactamases, and they will go on and just chemically cleave one of these bonds, and that disrupts the structure of antibiotic. And without the structure, antibiotic is just unable to perform its function. Okay, so these are beta-lactamases that do that. And one thing to remember this by is that this mechanism of resistance using the enzymatic inactivation is more common in gram negatives than it is in gram positives. So as it says here, occurrence in gram negatives is higher than in gram positives. As you can see here, thousands of different enzymes of beta-lactamases have been identified. Probably a lot more haven't been identified yet. They belong to more than 20 different classes. You don't need to know any of those classes just to give you the idea behind the diversity. And these kind of enzymes are responsible for degrading, for enzymatic inactivation of all sorts of beta-lactams, penicillins, the simplest ones, as well as cephalosporins and the carbapenems. These two classes, you must remember them from last lecture. Know that these two are beta-lactams. These are very important antibiotics in general for us, for humanity. Very effective antibiotics and very important ones. Not toxic. So as far as how it happens, like I said, it's just one of the bonds is getting cleaved, and antibiotic loses its functionality. These enzymes that I'm talking about, as per our earlier discussion, they can be encoded both on the plasmids as well as in bacterial chromosomes, so in the genomic DNA. So there's no particular one location for these enzymes. They can be both. They can be present both on the genomic DNA, so that would be innate motor resistance, and they can be also supplied on the plasmids, which is part of the phenotypic or acquired resistance. So what is the strategy for us, right, for the scientists to fight off these enzymes? So I don't know if I mentioned this in the last lecture, but this interaction between us and bacteria in terms of antibiotics production and application is commonly being referred to in literature as arms race. You know how they call it in politics, arms race between the countries. Why is that the case? Because that's literally how this relationship between us and pathogenic bacteria is going. They create the arms to kill us or to infect us. We create arms back to fight their arms, right? And then so bacteria creates all of these pathogenic factors to uh, infect us. We create antibiotics to fight that. As soon as they see antibiotics, they come up with, for example, the enzymes, right? To kill those antibiotics, to cleave those antibiotics. As soon as we see those enzymes, we come up with our own strategy. So it's like back and forth like this all the time. So what is our strategy in response to those enzymes? Um, what we do instead, we supply, we came up with a very efficient strategy, actually, make a note of that. Um, very efficient. It's not perfect. Bacteria, so another iteration of this arms race, bacteria does know how to respond to that, but still a very effective mechanism. So what we do in order to fight off these beta-lactamases, we supply the beta-lactamase inhibitors with the antibiotic. So now when you supply the antibiotic to the patient, you supply the antibiotic itself as well as the chemical that doesn't have any antimicrobial activity by itself. So it's not an antimicrobial of any kind. But what this chemical does, it is blocking or inhibiting the activity of the enzymes, bacterial enzymes. So a famous example for this would be the augmentin, which is basically a mixture of beta-lactam antibiotic, which is amoxicillin, and clavulanic acid. Clavulanic acid being the beta-lactamase inhibitor. So how does it work again? We supply this drug to the patient. Bacteria sees the beta-lactam and starts making beta-lactamases. But we have clavulanic acid in that formulation. And clavulanic acid inhibits the beta-lactamase. So if the beta-lactamase is inhibited, the antibiotic cannot be degraded. And therefore, amoxicillin can go in and actually do its job, right? Now, why is it a smart strategy? I told you it's a very good, very efficient strategy. Because the chemical that is actually doing all the job, all the important job, by important job, I mean the chemical that inhibits the enzymes, the ones that give us trouble, right? Because I mean, antibiotic does also a very important job by killing bacteria, by suppressing bacteria. But the key element here is the clavulanic acid. So this chemical doesn't actually inhibit or kill bacteria at all. Like it's not toxic to them, which in its turn means that bacteria doesn't recognize that chemical, the clavulanic acid, as a threat. Bacteria only responds efficiently to something that is directly killing it or inhibiting it. Clavulanic acid doesn't. It does so kind of indirectly as we can see, right? But bacteria doesn't have enough brains, so to say, right, to actually figure this out. So when bacteria faces this kind of a drug, what it does, it just starts making more of the beta-lactamases. But clavulanic acid can be supplied at pretty high concentrations, and all these beta-lactamases are being inhibited anyway. So as a result, the whole formulation works pretty well. Bacteria takes a while to figure out what exactly is responsible for, the negative outcome. So that's why the strategy is so efficient. However, like I said, in this arms race, as they call it, bacteria sooner or later figured out what's going on, and in response to these beta-lactamase inhibitors, certain types of bacteria, not all of them, but there are certain ones that are capable of producing these metalloenzymes. Metalloenzymes are basically beta-lactamases, the same kind of enzymes as beta-lactamases. So they are the ones that will cleave the beta-lactam antibiotic with one good property to them. From bacterial standpoint of view, these metalloenzymes are resistant to beta-lactamase inhibitors. So the clavulanic acid doesn't work against them, right? So again, it's like several layers of this arms race. Pathogenic factors versus antibiotics versus beta-lactamases versus clavulanic acid versus metalloenzymes. So like literally 5 layers. So that's a very, like I said, efficient strategy. Make sure you know how it works. Make sure what each component does. It's very straightforward. There's nothing really complicated here. So the actual chemical that kills bacteria, inhibits bacteria, is amoxicillin. Clavulanic acid is not toxic, as it says here. It has no antimicrobial activity by itself, the clavulanic acid. Amoxicillin does, but not the clavulanic acid. So that was one mechanism. So that was the enzymatic inactivation. Again, don't forget this is very important for the exam. This one is predominantly characteristic of gram negatives, specifically gram negatives. Now, gram positives have a slightly different strategy most of the time. It's not like they don't have the enzymes, but it's just enzymatic inactivation of the antibiotics is not nearly as common as the other method, which is the alteration of the drug target. It's a lot more common in gram positives than it is in gram negatives. So, what is this mechanism is about? In order to, I thought I had a link here. So there's a new, or it's not so new, you know already what it is, but you probably haven't seen this word before. So the abbreviation PBPs. So what are the PBPs? PBPs stands for penicillin binding proteins. That's what the PBP is. So what is that? We didn't encounter it last time. But you already saw them in the animation that I showed you. PBPs in this context is the same thing as the transpeptidases. And the transpeptidases, you should remember that from last week, are those enzymes that were responsible for establishing the amino acid crosslinks between the sugar subunits of the bacterial cell wall. Transpeptidases were the enzymes are the enzymes responsible for establishing those crosslinks, and therefore they are the enzymes responsible for structural integrity of gram positives and gram negative cells, mostly gram positives, because of the thicker layer of peptidoglycan in gram positives. So because of different considerations, because there are different types of enzymes present in the cell, and because of the ability of transpeptidases, or rather because of the ability of beta-lactam antibiotics to attach to transpeptidases, to bind to transpeptidases, right? That was the mechanism behind the beta-lactam mode of action. You were adding, if you were adding beta-lactams to the solution, they would bind to transpeptidases. Transpeptidases bound by beta-lactams were unable to cross-link the growing cell wall of a gram-positive cell, and therefore the cell wall was lacking the structural integrity, and as a result of that, the cell would be bursting open with any significant changes to osmotic pressure, right? So that's the mechanism. So in bacterial cells, gram positives, gram negatives, there's lots of enzymes, different enzymes that have this property. They're able to bind to penicillins or to beta-lactams. Remember, penicillins is the same as, well, not the same, it's just one of the representative of the beta-lactams. So what scientists did, they called all these enzymes that are capable of binding to beta-lactams as just penicillin binding proteins, with transpeptidases being a specific representative of that class. Okay, so now that you understand what the PBPs are, so alteration of the drug target in case with this mode of resistance is very straightforward. If you are, a bacteria and you don't want beta-lactam to act on you, according to this mechanism of resistance, what would you do? You would probably somehow change the affinity of these enzymes that beta-lactams are binding to actually bind to PBPs or to transpeptidases. That's exactly what they do. What they do is they mutate the transpeptidases in such a way that the penicillins or beta-lactams in general are unable to bind them anymore. As it says, modification of PBPs with transpeptidases being a specific representative of this class. So they modify these PBPs in such a way so that the binding of the beta-lactam drugs is not happening anymore. So they make them invulnerable to binding of beta-lactam drugs. Okay, and as a result of that, antibiotic cannot attach to those PBPs or the transpeptidases, and therefore it cannot inhibit the cell wall synthesis by the gram-positive cell. And if there's no cell wall synthesis inhibition, there's no antimicrobial activity. So as simple as that. So instead of like I gave you that analogy at the start, you have a key and you have a lock. The key is your antibiotic. The lock is your whatever cellular target of that antibiotic. So when antibiotic interacts with the cellular target, when the key goes into the lock, it unlocks the cell, right? Kills the cell. So one strategy is to modify the key, break the key, so to say. The other strategy is to break the lock or stick something into the lock so that the key doesn't go in anymore. So that's basically what this mechanism is making use of. They modify the lock, so to say. They modify the cellular target, which are the transpeptidases or the PBPs in a more general consideration. And as a result of that, your key, your antibiotic, is unable to attach to that target anymore. It's all structure function relationship, something that we discussed from week 1. Everything in molecular biology is structure functional relationship. If you have a change in structure, expect changes in function. If you have changes in function, the first thing you want to check how the structure was modified. Nothing is happening magically. It's science. It's a very mechanistic approach to a lot of things. So there's a very specific mechanism to all of these concepts. Things literally interact as if they were small objects, like engineering something, parts on a molecular level only. So we'll see more examples of that later on, even funnier examples. Actually, this one is actually a very funny example. So this one is the next class of antibiotics that we looked at last week, which were the glycopeptides. So these are antibiotics that work similarly to beta-lactams in a way that they also inhibit the cell wall synthesis. That's the similarity. But what's important to remember, they do so in a very different way. The cellular process affected is the same. It's the cell wall synthesis, but the mechanism by which this is happening is different. Okay. So major representative was vancomycin. Remember it. It was it's still very important drug. We're not going to go into that again. So how the glycopeptides were exerting their effect. They were blocking the cell wall synthesis by binding to peptidoglycan layer precursors. So the chemicals, the building blocks of the peptidoglycan layer. So if the antibiotic was bound to that precursor could not be used as a building block for the cell wall synthesis. Okay, so they were not binding to transpeptidases. These enzymes here. This was the case for beta-lactams. These ones do not care for the transpeptidases. Very important to remember. They were literally binding to the building blocks, as they call them. So the fact that they bind these antibiotics, the glycopeptides, bind to a completely different molecule. What kind of logical consequence in your head or what kind of logical take-home message you should right away generate from this? If it's a different mechanism, if it's a different molecule that is being impacted by these antibiotics, that means that the mode of resistance that worked in case of beta-lactams is not going to work in case of glycopeptides because it's a completely different molecule affected. Enzyme in that case, peptidoglycan layer precursor in this case. What is the mechanism of resistance to glycopeptides? It's actually a very interesting mechanism, very funny mechanism. So what one of those, there's two, I guess, we'll look at. So one, the one that I refer to as funny, is the second one, the piling up. So what bacteria does is a very simple conceptually method. What they do is they pile up, so they produce a lot of unlinked peptidoglycan layer precursors, the ones that are not going to be a part of the cell wall synthesis. Antibiotic doesn't know the difference. Antibiotic is just a molecule, right? It doesn't have any brains, right? So for the antibiotic, all it sees is a structure. And the structure of unlinked peptidoglycan precursor, the one that will not be involved in the cell wall synthesis, to antibiotic is exactly the same molecule that would be linked with peptidoglycan layer synthesis. So when bacteria switches on this second mechanism, they produce tons and tons of this unlinked peptidoglycan layer precursor. They therefore soak up, so to say, all of the antibiotic. So antibiotic gets not inactivated; it just gets soaked up by it attaches to this unlinked peptidoglycan layer precursor. But this unlinked one is not going to participate in the cell wall synthesis. So once the concentration of antibiotic goes down because of this method, the linked peptidoglycan layer precursor, most of it will safely go in and take a part in the construction of the cell. Okay, so a very interesting way. Like you can think of it, I usually give students the analogy of the sponge. It literally, these unlinked peptidoglycan precursors work as a sponge. Like you throw a sponge into the water and it absorbs all the water, right? Or whatever, most of the water. So that's what these unlinked precursors do. They soak up most of the antibiotic. It's never the whole thing. It's never 100%, but nevertheless, most of it is absorbed. Antibiotic obviously loses its effectiveness significantly because most of it has been just linked with precursors that are useless. They're not going to affect the synthesis of the cell wall. Um, some of you may say, well, what if we add more vancomycin, more antibiotic to counteract this, piling up of unlinked peptidoglycan precursor? Unfortunately, it's in most of the cases with antibiotics, and especially with vancomycin, it is not an option because vancomycin is a very effective drug, as we discussed, but it's also a fairly toxic drug. So you cannot just, prescribe infinite doses of vancomycin to a patient. You will kill the patient eventually. Vancomycin is not necessarily it's considered to be a drug of last resort, as we discussed last week, as I posted last week. Those drugs are pretty toxic. They are efficient. That's why they're drugs of last resort. They're efficient, or they can be efficient, but they are definitely toxic. There's consequences to that efficiency. So, and in general, antibiotics are, despite of selective toxicity, they are not necessarily the easiest molecules for our body to work with. So you cannot just infinitely increase the concentration. So that's the mechanism, though, the funny one, as I call it, whatever. Interesting one. The second one is a lot more straightforward, the one that is listed as first here on my slides. So what they do is what bacteria does is if you look at this structure on the right hand side here, the top one, this is the interaction of vancomycin, this huge molecule, right, with the peptidoglycan precursor under normal conditions. So vancomycin stably attaches to the precursor because of the multiple hydrogen bonds that are being formed between the vancomycin and the D-alanine residues of the peptidoglycan layer precursors. So you have multiple hydrogen bonds being formed, 1, 2, 3, 4, right? So because of these multiple hydrogen bonds, as you know from basic chemistry, hydrogen bonds are very good at, especially when there is several of them, they're very good at stabilizing different assemblies, right, in biology. After all, they're responsible for the DNA stabilization, right, between the base pairs. Uh, so when you have four of them, it's a very stable structure, and that's what makes vancomycin efficient. It kind of traps, if you will, the peptidoglycan precursor. So what bacteria learned to do is they realized that, well, they didn't really realize anything, obviously. I hope you understand the analogy. It's just a matter of error and trial over millions of years. So they came up with a strategy where they substitute the regular D-alanine links in their peptidoglycan precursors for serine and lactic acid. And these two amino acids have one fewer hydrogen bond, as you can see here at the bottom, with this major hydrogen bond here is missing. And as a result of that, this whole complex now between vancomycin and this modified version of a peptidoglycan precursor is much less stable, is much less stable. And because it's much less stable, vancomycin will, like when it's in a solution, right, when you have multiple of these molecules interacting with each other, this complex is not going to be stable, and as a result, most of these complexes will fall apart. They will fall apart, and that will obviously liberate the peptidoglycan precursor. And that peptidoglycan precursor can safely go in and take part in the production of the cell wall. So again, what are we looking at here? Structure-function relationship. You have a certain structure, you have a certain functionality in place. Vancomycin attaching strongly. You change the structure, you change the functionality. Vancomycin doesn't attach stably anymore, and as a result, the whole functionality of this antibiotic is lost, essentially, right? So that's how molecular biology works all the time. Everything has a very specific mechanism behind it. As long as you understand the mechanisms, you can understand and you can infer, you can predict a lot of things. If you don't understand the mechanisms, you will never understand molecular biology and science in general. It's one of the main messages I'm trying to pass to students. Must understand, must be able to answer the question why, how and why things are happening. If you understand that, you can solve a lot of cases. If not, every new case is going to be a completely new case to you, like whole new case to you. Resistance to vancomycin, speaking of, what's happening in their, how does the resistance emerge to these antibiotics out there? It will be the case for most antibiotics that we will look at. It's just excessive use of antibiotic. Excessive use of antibiotic, and vancomycin is no exception here. And also keep in mind that just generally across the globe, regulations on antibiotics have gotten stricter recently, but earlier it was much easier to get antibiotics without any prescriptions. So that also contributes to like overuse, abuse of these drugs, and as a result, unfortunately, the spread of resistance. So what were the major reasons for the emergence of the resistance? A very widespread use of oral glycopeptides to treat Clostridium difficile. We encountered this bacteria before. I told you it's the most common hospital infection. So glycopeptides were very widely employed to treat those infections. And even probably bigger source is glycopeptides, these antibiotics, not just vancomycin, but glycopeptides being used as feed additives to farm animals. And to wrap today's lecture up with this point, I don't know if I mentioned this last week, but when we talk about the spread of antibiotic resistance and about how we contribute to spread of antibiotic resistance, one factor that has always been kind of overlooked. Um, People talk about us abusing antibiotics in the clinics to treat people, but what people rarely talk about is that about 70% of all the antibiotics that we make as a society are the antibiotics that are being prescribed to animals, and only 30% of all of that mass of antibiotics is what we use for our own needs as people, as patients. So more than twice as much antibiotics are being prescribed to animals, farm animals. Why? Right? To keep them healthy so that we can get more meat, more milk, and whatnot from those animals. They're being fed antibiotics a lot. And that obviously also contributes to spreader resistance, especially on the farm. All those antibiotics are readily available in the soil. The rest is, what's happening, right? Evolution and environmental pressure from these antibiotics forces bacteria to mutate, 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 mutate. So they try to cut down on these quantities of antibiotics, but it's also a science in itself how much this approach is actually valid and how much is it actually being employed. It's not that easy. If you remove all the antibiotics, the prices of meat and all these dairy products are going to skyrocket. So let's wrap up with this 10:20. So we have about what 7 slides or 7, 8 slides remaining. They're not the most busy slides, so we will wrap them up next week during the first lecture hour of the lecture, and then for the last 2 hours I will take the questions. So come with the questions. Again, it's up to you. If you don't have any questions now that everything's been posted, it's not necessarily a big problem. But if you still have some questions, absolutely, the next week is the time to ask those questions. The last time to ask those questions, the last opportunity, including calculations. I'm assuming some people will have questions on calculations. Don't be shy. You can email me your questions if you want to stay like anonymous or something. But use that opportunity next week. This will be the last lecture next week. And then as soon as I know about the Thursday time on week 14, I will obviously post it. So keep your eye on that as well as in general on the announcements on Luminate. Okay. Alrighty, guys. So that's it for tonight. Have a good night. Have a good weekend, and I'll talk to you next week.